More than 300 butterflies are fluttering across Sottunga Island in the middle of the Baltic Sea. They appear during the short summer days, covering the coastal meadows with their delicate wings. Yet this peaceful scene is no coincidence. It began with an experiment in 1991, when Finnish scientists introduced caterpillars to the island and followed their fate. There was no genetic modification, no dangerous invasive species, just caterpillars, plants, and time. But after more than 30 years, the experiment has revealed a reality far more complex than originally predicted. Why was such a fragile population able to survive? And what truly happened on that small island in the Baltic Sea? Today, let's follow the journey of these caterpillars with Terran Works and explore the quiet ecological relationships that have shaped the fate of an entire island. Sottunga is located in the Åland archipelago, between Finland and Sweden, surrounded by thousands of small rocky islets rising from the cold waters of the Baltic Sea. From above, the island appears as a solitary patch of green amid gray waters, with a rugged coastline, barren rock outcrops, and low meadows stretching along the sea's edge. Sottunga covers only about 10 square kilometers and has a population of fewer than 100 people. There are no cities, no industrial zones, and no waves of mass tourism. Life here moves slowly. Narrow roads wind past scattered wooden houses. Winters are long and dark, while summers are brief yet filled with light. Ecologically, Sottunga is almost an ideal environment for observation. There are few people, little interference, and few uncontrollable variables. There are no large predators and no densely layered ecological networks like those on the mainland. Even the smallest changes can become clearly visible over time. It was precisely Sottunga's isolation and simplicity that drew scientists' attention. To them, the island was not just a quiet natural space, but an almost ideal environment for answering a fundamental question in ecology. How can a population survive when it is completely isolated? To explore that question, in the summer of 1991, Finnish scientists introduced caterpillars of Melitea synxia to the island. The caterpillars were divided into several small groups and released into meadow areas that had been surveyed in advance. On the surface, this action caused no obvious disturbance. There was no mass invasion, no immediate change that caught the attention of the island's residents. The caterpillars crawled slowly across the leaves, feeding, growing, and continuing their life cycle as if they had always belonged there. However, this was not a random decision. Each group of caterpillars was carefully documented. The release location, timing, weather conditions, and surrounding plant species were all recorded in detail. From a scientific standpoint, the experiment was considered controlled, at least within the limits of the variables that humans could observe and measure. Why choose butterflies rather than birds, mammals, or other insects? In ecology, not every species is suitable for long-term experimentation. Scientists selected butterflies for very specific reasons. First, the species does not migrate. Once born in a particular place, it will almost certainly live and die there. On a small island, this trait is especially important, as it allows researchers to define and control population boundaries without worrying about individuals leaving or new ones arriving from outside. Second, butterflies have relatively short life cycles. Within just a few years, scientists can observe multiple successive generations, directly tracking phases of growth, decline, and recovery, processes that might take decades to unfold in birds or large mammals. Third, butterflies are highly sensitive to environmental conditions. Even small changes in weather, vegetation, or seasonal timing quickly affect larval survival rates and adult population numbers. This sensitivity makes them particularly effective ecological indicators. Finally, the butterfly species Melitea synxia had already been extensively studied in the Åland archipelago for many years prior. The scientists entered the experiment with the confidence that they understood their subject well enough to anticipate most possible outcomes. Yet that very confidence created a paradox. When focusing too closely on a single species, it is easy to forget that no organism exists in isolation. The butterfly may have been at the center of the study, but it was only one link in a far more complex biological network. You know, transporting adult butterflies is nearly impossible. Their short lifespan, high sensitivity to stress, and tightly synchronized reproductive timing mean that even a small delay can cause an experiment to fail. For a study conducted on an island, that risk is simply too great. So instead of moving adult butterflies, the scientists focused on caterpillars in the diapause stage. Diapause is a special biological state in which development is almost completely paused. The caterpillars do not grow, do not pupate, and consume very little energy. This diapause state was artificially maintained by keeping the caterpillars at around 5 degrees Celsius, 41 degrees Fahrenheit, slowing their biological clock. Under these conditions, they could be stored and transported safely, without exhaustion and without entering the most vulnerable stages of their life cycle. Once removed from the cold environment and released onto Sottunga Island, diapause ended. The caterpillars were exposed again to natural conditions, began feeding on their host plants, resumed development, pupated, and eventually emerged as adult butterflies right where they had been released. When the first caterpillars were released onto Sottunga Island, their survival did not depend on the island's size, nor on human presence. It depended on their host plant, Plantago lanceolata. This plant is the primary and nearly irreplaceable food source for Melatea syncytia caterpillars throughout their entire larval stage. From the very first days after hatching, the caterpillars must find this plant. Without it, their life cycle would end within just a few days. Plantago lanceolata is not a rare species in the wild, but it is selective about its growing conditions. It requires abundant sunlight and open grasslands, and it struggles to compete once shrubs or dense vegetation begin to encroach. On islands like Sottunga, the survival of Plantago lanceolata depends directly on how grasslands are maintained over time. A reduction in grazing, or overly rapid natural succession, can allow woody plants to take over, causing the population of Plantago lanceolata to decline noticeably in a short period. What is striking is that the collapse of a butterfly population does not necessarily begin with parasites or disease. Sometimes the cause is simply a shortage of food. If Plantago lanceolata declines during a single growing season, large numbers of caterpillars may starve before they can pupate. There are no dramatic signs, no sudden catastrophe, just a quiet gap appearing in the life cycle of an entire population. Although Plantago lanceolata is the primary host plant, it is not the only option. Within Sottunga's ecosystem, another plant species plays an equally important role. Veronica spicata. This is a secondary host plant for Melatea syncytia caterpillars, and over the years, its presence has helped the butterfly population avoid complete collapse. The presence of Veronica spicata creates an important ecological buffer. When Plantago lanceolata temporarily declines due to unfavorable weather, localized drought, or shifts in grassland structure, Veronica spicata can serve as an alternative food source. This allows at least part of the caterpillar population to continue developing and survive harsh periods. However, this buffer is fragile. Veronica spicata is also a sun-loving species, dependent on open meadows and highly responsive to changes in land management. If both host plants decline simultaneously, even for a short time, the caterpillars are left with no alternatives. With limited mobility and no backup food source, the population can collapse at the larval stage. This leads to an ecological paradox. Sottunga may appear spacious and lush to humans, but for the caterpillars, their world is confined to the distribution of just a few specific plant species. Everything beyond that range, no matter how abundant, holds no survival value for them. Even when the grasslands were maintained, and when Plantago lanceolata and Veronica spicata were still present, the caterpillars on Sottunga were not truly safe. A few years after the experiment began, once enough long-term data had been collected to compare seasons, scientists began noticing something unusual. Many caterpillars were not dying from lack of food, nor from harsh weather. They grew, fed on leaves, and from the outside showed no visible signs of weakness. The population appeared to function normally. It was only when researchers began opening egg clusters and closely monitoring internal development that they discovered the presence of another organism, parasitoid wasps. These wasps are extremely small, entirely unlike the familiar image of wasps in everyday life. They have evolved to such a high degree of specialization that they parasitize only a very narrow range of hosts. In this case, their primary hosts were the eggs and larvae of Melatea cinctia. Female wasps locate egg clusters or young caterpillars and lay their own eggs inside them. The wasp larvae then develop silently within the host's body, using the caterpillar as their food source. The process leaves no obvious external signs until the caterpillar's fate has already been sealed. What surprised the scientists most was the efficiency of the parasitoid wasps on the island. Over many years of observation, the wasps seemed able to locate a large proportion of the butterfly egg clusters and maintain a stable presence over time. Even when the butterfly population declined and later recovered, the parasitoid wasps reappeared with remarkable consistency. The parasitoid wasps were not the final layer of the story. Beneath them, on Sottunga Island, an even deeper biological layer was quietly at work. It was Wolbachia, an intracellular parasitic bacterium that lives inside the bodies of insects and leaves almost no visible trace to the naked eye. Wolbachia does not live inside the caterpillars, nor does it kill them directly. But that does not mean it stands outside the story. In this experiment, Wolbachia was found inside the parasitoid wasps, the very species that directly determines the survival rate of the caterpillars. The bacterium is passed vertically from mother wasp to offspring, silently spreading across generations without ever needing direct contact with the final host. Rather than causing immediate death, Wolbachia interferes with more subtle mechanisms: reproduction, sex ratios, and the survival of the next generation of parasitoid wasps. Studies suggest that its presence can alter how parasitoid wasps interact with their environment, even making them more vulnerable to higher-level hyperparasitoids. When the parasite of the caterpillar is weakened, the pressure on the caterpillar population shifts accordingly. What makes Wolbachia so unsettling is its invisibility and quiet spread. There are no obvious symptoms, no early warning signs. Yet once Wolbachia enters a parasitoid wasp population that has never encountered it before, the entire balance between caterpillars, parasitoids, and the higher biological tiers above them can tilt in unpredictable ways. More than 30 years have passed since the first caterpillars were released onto Sottunga Island, and the experiment continues to be monitored with patience. The butterfly population has not disappeared, but it has never truly stabilized either. Numbers fluctuate over time, some years declining sharply, nearly reaching local extinction, then recovering again under more favorable conditions. What has drawn particular attention from scientists is that despite the island's small size, high isolation, and constant environmental pressures, the butterfly population has maintained a relatively high level of genetic diversity. At the same time, the parasitoid wasps and the bacterium Wolbachia have not vanished. They persist alongside the butterflies, tightly bound to the host life cycle, forming an overlapping, interdependent biological network that remains in a constant state of tension. Sottunga did not become an ecological paradise, but neither did it collapse into catastrophe. Instead, the island reveals a colder reality. Nature does not follow human intentions, whether good or bad. It does not reward, nor does it punish. It simply responds, slowly, precisely, and without mercy to the interventions we introduce. The story of Sottunga does not end in total collapse, yet it cannot be called an outright success either. It settles into an in-between state where every species still exists, but none stands on truly stable ground. An ecosystem that functions, but always under strain. This is not a story of scientific failure. On the contrary, the experiment was built on knowledge, data, and careful intentions. And that is precisely what makes it more thought-provoking. It shows that even when humans do everything by the book, control in ecology will always have its limits. What do you think about the story of Sottunga Island? Leave a comment below and subscribe to continue exploring future journeys with Terran Works.